Hello, everybody. It's Mr. Caudill. I hope you're doing okay. I certainly am doing okay. I like chapters 14 and 15. I think it's interesting to understand how we are who we are and how everyone we know got the genes that they have. And then you can apply this to all organisms. I have to say this before we move on. For years, I've been teaching this class for 20 years now, and for years we've been talking about recycling and pick up things that can be recycled because we want to save the environment, and the carbon dioxide levels are too high, and then mostly it's been the youth that have been latching onto this. And the point is that what we've been trying to do is prevent. We've been trying to prevent trash, prevent waste, prevent CO2 from getting in the atmosphere a little bit. We haven't really done a very good job up until this moment. I'm pretty discouraged, to be honest with you. And then last year, this came out. Finally, someone figured something out to be proactive. I always tell you, I believe in technology, and this individual at ASU should win the Nobel Prize. They finally figure something that's going to attack the problem. Now, I'm hoping that a few years from now, when you're watching this video, that we have 10 of these things, different types of inventions that are out there helping us get our carbon dioxide levels under control. I just have to say it. I don't think people think about it enough as of this moment, but I'm hoping that that will change. But let's, sorry, this is my platform. Let's talk about the chromosomal basis for inheritance. Now, chapter 15 can be really hard or long or whatever you want to call it. But if we just take it in chunks, and I'm going to show you what's important here. I don't want you to read the whole thing. I think it's definitely doable, but there are 4 or 5 concepts that you need to know for sure. Arizona State. Love it. Look at that tree. Okay, make sure that before you start reading for this chapter that you look at this. You got to know what I want you to read. Again, it's way too much if you just kind of plow into it. There's only 21 reading eye questions, and again, that's just like chapter 14. The reason why is because I want you to do some problems rather than talk about all the theory in the reading guide. Well, please remind your neighbor of the 2 Mendel's laws, law of segregation and law of independent assortment, because we're going to touch on those again here. Well, for the very first section of this chapter, I just want you to understand that there's this term called wild type. And what the wild type is the most commonly observed phenotype in the wild. So in this case, I guess let's say it's red eyes, or what flies usually have. That's the wild type. Now, you could have mutant phenotypes that have genes that are a little bit different. They express different proteins that express different characteristics. And in this case, it would be white eyes. That's it for the first section. There are a variety of different ways that animals determine gender in their offspring. We have the XYXX system. Remember, men have an X from mom, and then the sperm is what determines the sex of the child, because the sperm either carries an X or a Y. And so women have an X from mom, and then they have an X from dad's sperm. So it's really interesting, I think. And here's a movie on how some organisms determine their genders.
My wife is pregnant right now with our first child. And when people see her with her big baby bump, the first question people ask, almost without fail, is it a boy or is it a girl? Now, there's some assumptions behind that question that we take for granted because of our familiarity with our own human biology. For human babies, we take it for granted that there's a 50-50 chance of either answer, boy or girl. But why is it that way? Well, the answer depends on the sex determination system that has evolved for our species. You see, for most mammals, the sex of a baby is determined genetically with the XY chromosome system. Mammals have a pair of sex chromosomes, one passed down from mom and one from dad. A pair of X's gives us a girl, and an X and a Y together gives us a boy. Since females only have X's to pass on in their egg cells, and males can give either an X or a Y in their sperm cells, the sex is determined by the father, and the chance of producing a male or a female is 50-50. This system has worked well for mammals, but throughout the tree of life, we can see other systems that have worked just as well for other animals. There are other groups of animals that also have genetic sex determination, but their systems can be pretty different from ours. Birds and some reptiles have their sex genetically determined, but instead of the sex being determined by dad, their sex is determined by mom. In those groups, a pair of Z sex chromosomes produces a male. So these males only have Z's to give. However, in these animals, one Z and one W chromosome together as a pair produces a female. In this system, the chance of a male or a female is still 50-50. It just depends on whether mom puts a Z or a W into her egg. Certain groups have taken genetic sex determination in completely other directions. Ants, for example, have one of the most interesting systems for determining sex, and because of it, if you are a male ant, you do not have a father. In an ant colony, there are dramatic divisions of labor. There are soldiers that defend the colony, there are workers that collect food, clean the nest, and care for the young, and there's a queen and a small group of male reproductives. Now, the queen will mate and then store sperm from the males, and this is where the system gets really interesting. If the queen uses the stored sperm to fertilize an egg, then that egg will grow up to become female. However, if she lays an egg without fertilizing it, then that egg will still grow up to be an ant, but it will always be a male. So you see, it's impossible for male ants to have fathers. And male ants live their life like this with only one copy of every gene, much like a walking sex cell. This system is called a haplodiploid system, and we see it not only in ants, but also in other highly social insects like bees and wasps. Since our own sex is determined by genes, and we do know of these other animals that have their sex determined by genes, it's easy to assume that for all animals, the sex of their babies still must be determined by genetics. However, for some animals, the question of whether it will be a boy or a girl has nothing to do with genes at all, and it can depend on something like the weather. These are animals like alligators and most turtles. In these animals, the sex of an embryo in a developing egg is determined by the temperature. In these species, the sex of the baby is not yet determined when the egg is laid, and it remains undetermined until sometime in the middle of the overall development period when a critical time is reached. And during this time, the sex is completely determined by temperature in the nest. In painted turtles, for example, warm temperatures above the critical temperature will produce females within the eggs, and cool temperatures will produce a male. I'm not really sure who came up with this mnemonic, but you can remember that. When it comes to painted turtles, they are all hot chicks and cool dudes. For some tropical fish, the question of will it be a boy or will it be a girl isn't settled until even later in life. You see, clownfish all start out their lives as males. However, as they mature, they become female. They also spend their lives in small groups with a strict dominance hierarchy where only the most dominant male and female reproduce. And amazingly, if the dominant female in the group dies, the largest and most dominant male will then quickly become female and take her place, and all the other males will move up one rank in the hierarchy. In another very different ocean animal, the green spoonworm, the sex of the babies is determined by a completely different aspect of the environment. For this species, it is simply a matter of where a larva happens to randomly fall on the sea floor. If a larva lands on the open sea floor, then it will become a female. But if it lands on top of a female, then it will become a male. So for some species, the question of boy or girl is answered by genetics. For others, it's answered by the environment. And for others still, they don't even bother with the question at all. Take whiptail lizards, for example. For those desert lizards, the answer is easy. It's a girl. It's always a girl. They are a nearly all female species, and although they still lay eggs, these eggs hatch out female clones of themselves. So will it be a girl or will it be a boy? Throughout the entire animal kingdom, it does really all depend on the system of sex determination. For humans, that system is a genetic XY system. And for me and my wife, we found out it's going to be a baby boy.
Yay. Scientists believe that this is what happened with the dinosaurs when the KT event occurred, and that asteroid the size of Mount Everest hit the Yucatan Peninsula 76 million years ago, and 67 million years ago, and all of this dust kicked up in the atmosphere. The sun was blocked out for about 2 years, or maybe 3 years, they believe. They can see if you mine down across the world to a certain level of 67 million years ago dirt or rock, then all of a sudden you see this one layer there. It's fascinating. If the plants started to die, you know that there would be very little oxygen and very little food. It must have been an incredible selective pressure on all organisms surviving. But the point is the reason why the dinosaurs had trouble, scientists believe, is because they had this kind of reptilian sexual determination where the sex of the offspring wasn't determined until midway through kind of the gestation period in the egg. And the point is that it got cold. And so hot chicks and cool dudes, so there was all guys running around, dinosaur guys. There were no females around. If you've ever been to the tropics and you've seen like a turtle hatchery, what they always try to do is keep the eggs cool. So the scientists or volunteers in the middle of the night will watch turtles come out of the ocean. They'll watch turtles lay these ping pong sized balls into these sand pits. I've seen this and in Costa Rica, and then it'll be covered up, and then they'll basically protect the pit from, they don't want like an animal in there eating the turtle eggs. Now, of course, in nature, they shouldn't be doing this, but these are like conservatory places. And the point is they always put a shade up over it because they want females, because it's just like hunting around here. You can kill a lot of the guys, and one guy can impregnate 30 females. So females are where it's at. I think probably all of you already know that. But females are important. Let's keep going here. Sex-linked genes inhibit unique patterns of inheritance. We'll get into sex-linked determination in just a second. But there is a difference. So all of our chromosomes are the same, generally speaking, except for minor tweaks, which give us the different eye colors that you have and someone else has in the room, or the hair color or the height or whatever. But generally speaking, you have a gene that creates a protein that creates eye color in the same place, essentially. It's all the same for every chromosome and every person except for these sex chromosomes. The X chromosome and the Y chromosome are super different than one another. I've written up here, size does matter. The X chromosome has 306 million letters, and it has 2,000 genes. I would say most of which don't have anything to do with gender determination. The Y chromosome, by contrast, has only 78 genes, and half of these genes deal with stuff down in the testicle of guys that makes males males. The problem that we have here is that when you have a man and you have these two genes and you have then a lowercase letter here, let's make a lowercase b. Let's say this is a recessive gene that creates a protein that doesn't work as well as another individual. The problem is that when you have a guy in all of his cells, he doesn't have anything here. There's no chance for him to have a big B and have that be able to dominate over this poorly efficient protein that's going to then cause some sort of problem, some symptomatic problem, and someone says, oh, you have this problem. But if you're a female, so let's do this. So if you're a female and you have a lowercase b, here's the good news. You have another copy here. And this could then dominate over this, and you would never even know that you have this recessive gene here. This is why men have more diseases than women do, because of this fact. This thing is literally smaller. The sperm that create male babies literally weigh less. It's like 10% less than the ones that create female babies. And they swim faster, but like men are young men in the world, and I was one of them once, they burn out more quickly. And then the sperm that contain an X chromosome, slower, more deliberate, but stronger. Whatever. Okay, I'm just going to stop now. You might ask yourself, well, if someone is a girl, they literally have more genetic information, and how do we know which X that is turned on in order to create proteins? You get the X from your mom or the X from your dad that dominates in every cell. The answer is, you're going to read it in the book, is that in embryonic development, for each cell, one of the X chromosomes actually crumples up into like a ball, and it's called a Barr body. And so this is determined by random. And so let's say it happens when there's 1 million cells in the developing embryo of a female baby. At that moment, all of the X chromosomes, it's randomly determined, and then just one of them is going to work in each cell, and it could be the one from that little baby got from mom or from dad. I think it's kind of interesting. The X chromosome has genes for many characters unrelated to sex. A lot of characters related to like heart development and vision development are on the X gene. For a recessive sex-linked trait to be expressed, a female needs 2 copies of it, and a male only 1 copy. You can imagine that in order for a female to express this negative symptom, she would have to have a little b and a little b. So let's do this. Because color blindness, along with hemophilia, muscular dystrophy, Fragile X syndrome, these are these sex-linked syndromes that exist, and they exist more often in men than women. Men have color blindness. About 8% of the North American population has it. And we're going to run through this scenario here because you're going to have to understand how to do this on the test. So let's imagine that mom has 2 X chromosomes, and let's imagine that she's a carrier of this gene that causes color blindness. Maybe it's a gene that has a misshapen protein that doesn't allow the cone, it would be the cones in the eye in order to see red or green. For whatever reason, she just inherited it from her ancestors. So let's give her let's do it in a different color. Let's give her a big C and a little c. Now dad's gonna have an X and a Y, and so let's imagine that dad is able to see color. So let's put them here. So dad is able to see color, and mom is a carrier. Go ahead and do this. Press pause and do this Punnett square cross on a separate piece of paper, please. So here's what we see. So sorry, mom's a carrier, right? So she has a big C and a little c. So here's what we're seeing here. So any girl that this pairing of parents has is going to be able to see color. 50% of the boys that are created here are going to see color, but 50% of the boys are going to be colorblind, even though neither parent was colorblind. And you can see then the power of these recessive genes when it comes to sex-linked chromosomes in determining whether or not individuals have certain diseases or not. Because what this suggests then obviously is that this gene for the ability to see red or green exists here on the X chromosome, and then the males don't have it, like a corollary there, something to kind of offset maybe a recessive gene. We had a student a few years ago, and then his whole family came through the school, 3 kids. And he told me that his grandfather had a super recessive condition of the inability to see gray or blue. It was called gray-blue color blindness. And what happened in the Vietnam War was the government went through all these census data, and they figured out kids, young kids, let's say 18 years old, who weren't able to see gray or blue, had this condition. And they realized that they were very able to pick out patterns well. And so they put these young men on the back of planes, and they flew patterns over Southeast Asia and Laos and Vietnam. And what they were looking for was breakups in the forest canopy layer. And they were looking for camouflage netting that would describe Viet Cong installations down there. And so they would, apparently a normal person would just look and they would see the forest canopy, and they maybe not be able to see the breakup of the camouflage netting over maybe a gun position. And these individuals would be able to see it from a long distance away and say, that's where the enemy is. And then we would go fight the enemy. Let's watch a little movie on red-green color blindness.
Fans didn't respond too well to the NFL's new Color Rush campaign during Thursday night's Jets-Bills game, especially colorblind fans. Some colorblind viewers were unable to distinguish between the Bills' red uniforms and the Jets' green uniforms. Lawrence Tynes, former Giants kicker, was one of those fans who wrote on Twitter, quote, "If you're colorblind like me, this NFL game is going to be hard to follow. Football is America's sport, and a recent estimate suggests that 10 million Americans are colorblind, and the most common kind is red-green colorblindness."
While observing fruit flies, Morgan discovered a male that had white white eyes instead of the typical red ones. He mated the white-eyed male with a homozygous red-eyed female. The entire F1 generation, whether male or female, had the dominant red eyes, just as Mendel would have predicted. However, the F2 generation held a surprise. Just as Mendel would have predicted, 3/4 of the offspring had red eyes, and 1/4 had white eyes. But there was a catch. Only males had white eyes. 1/2 of the males had red eyes, and the other half white eyes. If sex and eye color were being inherited independently of one another, as Mendel would have predicted, then 1/2 the white-eyed individuals should be female. Obviously, eye color was somehow linked to sex. In order to explain his results, Morgan hypothesized that while there is a gene for eye color located on the X chromosome, there is no corresponding gene on the Y chromosome.
Okay, go ahead. Turn to your neighbor and explain sex-linked diseases and why they're more common in males, please. Well, chapter 15 goes really quickly. You already know about linked genes. They tend to be inherited together because they are located near each other on the same chromosome. We already did this in the meiosis chapter. So this is kind of a review. Make sure that you only read the sections that I'd like you to read. Linked genes are not independently assorted. We already talked about that in chapter 14, that law of independent assortment. That's it for 15.3. But here's what's important is that I get it that genes can be linked together, maybe ones that are closer to one another towards the end of chromosomes, which means that they get passed together during crossing over and we can follow them and then the resultant production of eggs or sperm, no problem. But what we then can do is by measuring how often genes are linked together as a result of looking at the phenotypes in the offspring for certain characteristics, like I told you the other day, red hair and freckles and blue eyes, we can then create a map and we can say how often these things recombine together. And you know, I wouldn't want to do this, but there have been really smart scientists that have done this, particularly this guy, Thomas Hunt Morgan, who did it like, 100 years ago when he didn't really have much technology at all at his disposal. It's pretty impressive. He did it at Columbia University, and he was able to map or put a gene map or figure out where the genes were for all sorts of things that govern characteristics in fruit flies. Here's a 1-minute movie.
Morgan and his students used this relationship between the distance separating two genes and the chances of a crossover splitting them apart to figure out where on the chromosome these genes are located. First, through crossbreeding experiments, they discovered a number of genes that were linked together in a series of pairs. They were therefore all on the same chromosome. Then, through further matings, they tested each linkage for its tendency to break, which told them how likely it was for a crossover point to fall between the two genes. From these numbers, they were able to work out the relative distance between each pair of genes. Through a simple puzzle-solving exercise, Morgan and his students were then able to line these genes up in their proper sequence and distance from each other. They had created the first gene map of a chromosome.
The good old days when people used to wear suits to school or to do lab work, or whenever they went on a plane, they would wear their best suit. I certainly wasn't alive during those times, but some people miss those times, simpler times. So we're already at 15.4. Alterations of chromosome number. Now, remember, we're talking about chromosome number here. How many chromosomes are in each one of our cells? How many? 24, 46? 46, right? 46 chromosomes. 2 sets of 23 chromosomes. 1 set from mom, 1 set from dad in each one of our cells. We can have alterations in those chromosome numbers. We can have 47 chromosomes in a cell, or as you'll see with polyploidy, we can have a whole extra set of chromosomes. Here's how this happens when it comes to just 1 or 2 fewer or 1 or 2 more chromosomes than the normal number of 46 chromosomes. Well, remember, we're dealing with meiosis here, And what's happening here is we have something called non-disjunction. Say that with me, non-disjunction. That just means that things don't disjoin when it comes to anaphase 1 of meiosis 1. The non-disjunction are these homologous chromosomes. There's also non-disjunction later in anaphase 2. But if in non-disjunction, pairs of homologous chromosomes don't separate normally, and then you can imagine if you have maybe one extra chromosome, it's going to then go and there's going to be a second division, and now you have a sperm that has an extra chromosome in it. Here it is, a number of chromosomes in the bottom left there, non-disjunction of homologous chromosomes in meiosis 1. You can see that those two sperm cells have either one extra or one few or too few chromosomes, because the whole homologous chromosome pairing went over to one side incorrectly. Well, this can also happen in meiosis 2. And when that happens, you just get 1 out of the 4 eggs or sperm with an extra chromosome, because this is a non-disjunction of sister chromatids in meiosis 2. So there's a difference here. Make sure that you understand this difference and the ramifications of that. This is just anaphase that's occurring here, and you can see that there was non-disjunction and that there was an extra chromosome that kind of went over accidentally to one side. Let's watch it. Oh, let's not watch it again. Here's my cartoon.
Do you know anyone with Down syndrome or Klinefelter syndrome? These are examples of chromosomal disorders, which are due to non-disjunction. When non-disjunction occurs during meiosis 1, replicated homologous chromosomes do not separate correctly. One daughter cell retains both replicated chromosomes. The other daughter cell lacks that chromosome. When a haploid sperm fertilizes an egg with an extra chromosome, trisomy occurs. When a sperm cell fertilizes an egg that lacks a chromosome, monosomy results. In this scenario, either a trisomy or a monosomy results. Non-disjunction can also occur during meiosis 2. In this scenario, meiosis 1 occurs normally. When the daughter cells separate during meiosis 2, one daughter cell divides normally, but the other daughter cell divides abnormally. One egg lacks a chromosome while the other egg has an extra chromosome. When a haploid sperm cell fertilizes a haploid egg, a normal diploid results. If the sperm cell fertilizes the egg that lacks a chromosome, then monosomy results. If the sperm cell fertilizes the egg with an extra chromosome, then trisomy results. In this scenario, there is a 50% chance of a normal diploid, a 25% chance of a monosomy, and a 25% chance of a trisomy.
All right, so most commonly, the chromosome which gets carried over through non-disjunction, which leads to a viable pregnancy and then a baby being born, is the smallest one. And you can see an example here of Down syndrome. This is happens in 1 out of 700 U.S. kids, and you can see that there's 3 copies of chromosome 21. Now, you have to believe, I believe that this non-disjunction would occur with all sorts of chromosomes, chromosome 1, chromosome 2, chromosome 3, but you can imagine that it's just basically TMI, too much information, I think, then gets into one cell, or not enough information in the other gamete that goes on to create a baby. And then the gestation doesn't go to term. 75% of all pregnancies that occur in North America actually are spontaneously aborted, often within the first few days. A woman doesn't even know that she was pregnant. And the reason why, I believe, quite often is because there's this non-disjunction of maybe chromosome 8, that it's much bigger than chromosome 21. For some reason, we're able to survive, humans are, if there's a trisomy of chromosome number 21. The frequency of Down syndrome increases with the age of the mother. I'm just reaching over here. You've seen these karyotypes before. There's one on the bottom right side of your picture there, but this is a colleague of mine here at Battle Mountain, and they got a karyotype of cells of their unborn child just to see if there was an alteration of chromosome number, if there was any trouble there. Just understand that the incidence of trisomy 21 increases with the age of the mother, increasing over 4. Well, there's other syndromes as well when it comes to non-disjunction. And if we have non-disjunction of the sex chromosomes and we have an extra or not enough sex chromosomes, we can get different syndromes. Klinefelter syndrome is a result of an extra X chromosome in a male producing XY individuals. These individuals have no facial hair and minimal breast formation. There's another one called Jacobs syndrome, which is XYY. These are called super males, is a term that I heard once. These individuals have more testosterone production and more things that you think make men men. I had a student a few years ago, and he stood up in front of class and he said, I have Jacobs syndrome. And he said, I played for the Husky football team, and he had, what was interesting was he had like way more facial hair than a normal, maybe 18-year-old, or more acne, things that you would think, and he was very strong. You could tell he had a lot of muscle. Things that manliness, that you would say, oh, that's manliness, right? Or whatever, make a guy. And he played saxophone in our band, and he played football. And I remember he said that he was a lineman, and whenever he got angry or someone was not playing fairly or whatever, he would get pissed off, he said, and he would just feel the testosterone raging through his body. And he said he got so strong at some points in the game that he could pick up other players and throw them to the ground. I found that to be very interesting. Turner syndrome occurs, I think there's a statistic in your book, I forgot the prevalence of Turner syndrome. These are females who have one X and then no sex chromosome from the other parent, so they just have an XO, and you can see that there's some characteristics that individuals with Turner syndrome has. There's another one called trisomy X, and these are females that have an XXX. They have 3 X's. This usually happens in about 1 out of every 1,000 female babies born, and they're often much more slender than other females. But of course, there's many people who are slender, so I don't know. So anyway, you can read all about this. That's just something I've memorized. You can read about all this in your textbook if you'd like. Well, phenomenon called polyploidy is real interesting, and you can imagine how rare this must be. Instead of just having one extra or one too few chromosome, we're talking here that you have an entirely extra set of chromosomes. So in this case, what would that be? Another 23 would be 69. So that would mean like you would have a cell with 69 chromosomes. I mean, I don't know if that really would ever happen in a human. This phenomenon of polyploidy happens often in plants, and you'll have plants that are living right next to other plants that have a full extra set of chromosomes. Bananas have an octoploidy number, or 8N, 8 sets of chromosomes per cell. Bananas have 3, they're 3N. Wheat is 6N. Super rare, again, in animals. Very common in plants, and the only animal we've ever found is this type of rodent. It was a type of field mouse that has 3 sets of chromosomes. In fact, I've read that the sperm that goes to create the baby of these animals has a super large head on it. I'll put my hands away, because you can imagine it has evolved to be able to swim that or facilitate that extra set of chromosomes down the fallopian tube of the female. Pretty crazy. Go ahead and turn to your neighbor. We're almost done. Tell them what you know about these conditions, please. What you feeling like this? It's late in the semester. We're almost done. Okay, sometimes chromosomes can actually break, literally break. Imagine a chromosome. Imagine a breakage of a chromosome. We're talking about millions of nucleotides. Would that be a problem? Quite often it is a problem. There's different types of breaks. Here they are up here. I'll move out of the way. Here's the first one, deletion. You can imagine if you delete a portion of a chromosome, that will lead to a lot of trouble. There's a common deletion which occurs on the chromosome 5, and I don't know why. And individuals who suffer from this deletion often have limited mental development, and their cry is like a meow. So you can imagine, what does that section of chromosome 5 that might be a little more fragile than other sections of chromosomes control? Well, the sounds a baby makes when they're upset, and the brain development. So this is called cri du chat, or cry of the cat in French. You can also imagine that if you duplicate a section of chromosome, again, too much information might be the result, and that might be a big time problem.
So imagine this yellow portion is the portion where the trinucleotide repeat is. And as you know, helicases open the DNA and DNA polymerases copy it. So we're going to watch the repeat now. Get through the helicase, this single strand is shown. The DNA polymerase slips on that repeat and a hairpin is formed. And as this now will go through, you will watch as we go through the second cycle, next round of replication. This longer repeat will now get replicated and incorporated in our DNA. And you're going to end up with an expansion. And here you've seen a small extension, but imagine this going on again and again and again. So with time, this will continue to get bigger, and that's why the disease will keep getting worse.
Okay. Let's talk about inversion. You can imagine that a segment within a chromosome, when it's flipped around, that might be a problem. I'll show you why in a second.
An inversion is a reversal in the order of a segment of a chromosome. Inversions do not alter gene expression unless the breakpoints on the ends of the inversion occur within a gene. When a segment of a chromosome is inverted, it can pair in meiosis only by forming an internal loop. Although crossing over occurs in the 4-strand stage during meiosis, only 2 strands are shown for clarity. Crossing over that occurs within the inverted segment during meiosis can result in some genes being lost on one chromosome, while others are duplicated on another chromosome.
Okay, here's the last one, translocation, where a portion of a chromosome is literally broken off and moves to another. Now, you can imagine this happening randomly, but for some reason it happens more commonly in between certain chromosomes, like the 420 translocation, there's a 1421 translocation, there's a 922 translocation that happens. Here it is. More common than others, and I don't know why that is. I don't know why those bits of chromosomes are more fragile in terms of breaking off and then reannealing, not at a random chromosome, but at the same one again and again. And we see these. In this instance, you can imagine that the portion of chromosome 9 that breaks off here is responsible for cell division, management of cell division. So that could cause a type of blood cancer called chronic myeloid leukemia. Well, why might these spell trouble for transcription? And I've drawn this up here. We've seen this before. Just remember that in order for, so I'm just going to write TF up here for transcription factor. In order for the RNA polymerase to jump on and do transcription of any sort of segment of gene, remember, way upstream up here, not only does this thing have to bend over and cause it to pop off and allow the RNA polymerase to go, but where are these transcription factors made from? Well, they're made from these genes that are up here, and there's all sorts of proteins that then turn on those. Those are called enhancers, and we're going to learn about all this in chapter 18. But the point is it's super, super choreographed, and if you have any sort of problem here where you're inverting things or duplicating things, you can imagine that this wonderful kind of chemistry that has to happen here, all in a sequential fashion and all under control, will be thrown off. And so that's the problem.
The minimal importance of how a simple code was turned.
I'm just showing you this again because I want you to be able to visualize what I've drawn up here on the board. You can see that there's at the beginning of a gene there's some transcription factors that are sitting there. There's probably a repressor protein that's also sitting there saying, all right, no one can do transcription on this gene and make a protein from this message until I get popped off here. Again, it's super, super controlled. These all proteins have to be in the correct shape. They have to be in the correct order. You can see that they're all coming here. They're probably all coming from upstream. And if you snap off of a chromosome and you put it on a whole different chromosome, some proteins will be made out of order, and you can sense how this would not be done well after that. Okay, if you were in class, I'd give you some time now to work on some problems here, but otherwise, thank you. We're done with chapter 15 as far as lecture goes. Thanks for your attention.